You're watching CTOM TV. Knowledge is power.
The DNA spiral contains these cross-links. So the DNA is basically two strands, one on the outside of each side. And then there's these cross-linkings. And so each one of those cross-linkings is referred to as a nucleotide or a DNA base pair. And so what happens is there's four of those, and they're basically named based on the type of amino acid that is attached to it. And so you have what we call the G for guanine, the C for cytosine, the T for thymine, and the A for adenine. And so it's the order of these A, G's, C's, and T's that decide what the gene product looks like. So a mutation, could you go back, please?
Sorry, I didn't mean to do that.
Yeah, so it's the order of these nucleotides that determine the gene structure. And a mutation is anything that changes it. So in other words, if you have a T in that middle position there and it gets mutated and turned into a G, then that's going to change the function of that gene. And that gene can become from a normally functioning gene, it can become a gene that now causes cancer or causes the cells to grow and divide on a regular basis. So what genetic testing is, tumor DNA testing is it looks at approximately anywhere from 300 to 500 different genes is what we typically use now. And it looks at each one of these individual base pairs and determines whether they've been mutated and changed or not in your cancer cells. And so what's important with tumor DNA testing is that even one base pair change can radically change how that gene functions. So it's really important to know that. So in order, you may think, okay, well, that's not a big deal, just do some simple genetic testing. But the amount of possible mutations that can occur is staggering. If you think, I like to use astronomy as an example. I mean, we look up into the sky and we see a little tiny dot, and that could be billions of planets and stars, that one little dot. Genetics is much more complicated than that. Just for example, your standard gene can be anywhere from 10,000 to as big as 230,000 of these little base pairs. That's a lot. So if you have even 150 base pairs, in order to determine how many different possible mutations can occur in even 150 base pairs, you would take the number of nucleotides that can possibly occur at that one position, and that would be 4, and then you would put it by the power of 150. And so even just 150 nucleotides, which is a tiny, tiny little portion of even a smallest gene, that number, 4 by the power of 150, gives you a number that is larger than all of the atoms in the known universe. I mean, that's a staggering number of atoms. I mean, just on the planet, the number of atoms is huge, but all of the atoms in the known universe, just in a tiny portion of a piece of DNA of a gene.
So bring this back to us. How does this affect us if we have cancer?
So if you have cancer, it's going to tell you that every person's cancer is 100% unique to them. No two people on this planet have the same cancer.
Mm-hmm. Because it's based on those mutations.
Because everyone has their own set of unique mutations. Yeah. And the chances of two people on this planet having the same genetic mutations together is unheard of, even in identical twins.
Mm-hmm. So back to talking about the genetic testing, there's 3 different types here. Would you like to tell us, you've told us a little bit about tumor DNA testing. Mm-hmm. If you feel like recapping that, please do, but tell us more about these other 2.
So so tumor DNA testing has been around since about 2005, when we first did the Human Genome Project. The goal then, I remember, because it was a big deal, is like, wow, I can't wait until we can do one genome for $1 million, then things will really change. Now you can actually do tumor DNA testing for anywhere from $5,000 to $10,000. I mean, it can be as high as 30,000, depending on how many genes you look at. But there's many commercial tests available. Michelle, how many tumor DNA sequencing companies are out there right now? Do you know?
Well, I don't know about companies, but I know that the most current statistic I saw for a couple months ago says that there are 7,000 different types of genetic tests right now,
Wow.
all specific to
Wow.
cancer, but many are.
Wow. That's just incredible.
Yeah, it's a lot.
So tumor DNA sequencing, very important for anyone with cancer. There's also a new test that's just been, I mean, we've had it in the lab, we've had it in research for many years, but now it's become cheap enough that we can actually do it for patients. And it's called RNA expression testing. And so RNA is the intermediate between a gene and a protein. It's a way of measuring how much of that gene is being produced. So when a gene gets turned on, it gets turned on briefly, it creates an accumulation of a protein product, and then that gene gets turned off. RNA expression testing allows us to determine how much of that gene is produced. So not all cancers are caused by genetic mutations. I mean, yes, they are, but they're genetic mutations in genes that control the expression of the genes. So, you know, it gets very complicated. So rather than having the gene mutated, some cancers have what we call epigenetic changes. And those epigenetic changes are changes in how much of that gene is produced. So the DNA in all of our cells is the same in every single cell. It's not exactly the same because there's a lot of stochastic processes going on, but theoretically, in a general term, it's the same. The way we produce different tissues and the way we are able to become a multicellular organ is that in some tissues, certain genes are turned on, and then other tissues are not turned on. So we actually have the DNA to grow wings in our body, but it's just not turned on. It's turned on in a bird, but not in us. So epigenetic testing or epigenetics is the process of turning on these genes and turning off these genes in different cells. So what happens in cancer is certain genes get turned on and turned off at different levels. And RNA expression testing is a way of testing for that and seeing what genes your cancer is over or under expressing compared to your normal tissues. So the test we like to use is an RNA expression test. It looks at 20,000 different genes in your tumor. And it says these genes are overexpressed and these genes are underexpressed and compared to your normal cells. And it compares it to two types of normal tissues. So it's an amazing new tool. And then it uses an artificial intelligence platform to match those overexpressed drugs or overexpressed genes with certain drugs. So there's also inherited mutational testing, which is probably what most people understand as genetic testing. And it basically just looks at genes that are potentially inherited. And so typically you're not going to look at more than about 100, 150 genes that we typically follow. Cancers that are caused by inherited mutations do not occur as often as we used to think. I think it's about 5 to maybe 10% of people with cancer have it inherited. Most of the cancers are what we call sporadic or somatic. In other words, these mutations happen over our lifetime in a certain group of cells based on exposure to radiation, even oxygen. Oxygen is a horrible thing for mutating DNA. You know, smoking, any sort of carcinogen exposure. A carcinogen is anything that mutates DNA and changes the structure of a gene.
Thank you for watching CTOME TV. Subscribe below and stay informed.